welcome back all. We are really going to dive into the adaptive immune response starting with this lecture. We're going to talk about cell immunity, which is the function of T helper and cytotoxic T cells in the immune response. And we're also going to talk about natural killer cells, which are part of the innate immune response, but this is a really good time to bring them up. Just to preface that this is probably going to be a little bit longer than some of my previous lectures because there's a lot of information to talk about here. I'll do my best to organize and to explain it as thoroughly as possible, but please feel free to stop, take some notes, take a break while you're watching this because it could get a little long. T cells, both T helper and T cytotoxic T cells, are made in the bone marrow and they are selected within the thymus. The bone marrow here is shown on the bottom left of the picture. In this particular case, we're looking at the bone marrow of the femur, which is a really big site for production of blood cells, both red and the white blood cells. Actually, this is the location of the hematopoietic stem cells that we talked about in previous lectures. The thymus, you can kind of think of as like the training ground for T cells. In the thymus, T cells are exposed to a variety of self and non-self antigens. And really what's happening here is that the T cells, each one that's being produced in the bone marrow, is being selected or tested to determine whether it will respond to non-self or foreign epitopes, but not respond to self-epitopes. In other words, what's really being asked here by the body is the T cell receptor that is going to be important for the T cell to recognize foreign antigens, is that particular receptor functional? After T cells have been selected, they will travel throughout the lymph system, making about one complete round of the lymph system every 24 hours. And as it's passing through the lymph nodes and spending time in them, T cells are going to be interacting with professional antigen presenting cells, determining whether they can detect a pathogen or not. There are three major types of T cells known as T helper, T cytotoxic, and T regulatory. Each of these has a very important role in modulating or regulating the immune response. The first thing that all three of these cells have in common is they have what's known as a T cell receptor. This T cell receptor will recognize a specific epitope of a pathogen. Next, they all have what's known as a co-receptor. CD stands for cluster of differentiation, of which there are many different types, but these are all kind of, we think of as cell markers or cell receptors that carry on functions within the cell. Particularly, T helpers and T regulatory cells have a co-receptor known as CD4, whereas those cytotoxic T cells have a co-receptor known as CD8. And we'll come back to the role of these co-receptors in a little while. T helper and T regulatory cells are both activated by the presence of an epitope on MHC2, whereas cytotoxic T cells are activated by epitopes, presented on MHC1. Finally, there's a difference in the function of each of these three. We often think of T helper cells as orchestrating the immune response, both the adaptive and the innate immune response, and we'll talk about how it does that function. We also know that cytotoxic T cells destroy infected cells, and finally, we've begun to understand more and more how T regulatory cells are involved in what we call immune tolerance. Immune tolerance is in essence, the ability to control the immune system from responding to the host. In other words, T regulatory cells have a very important role in preventing autoimmune diseases. Now, as already mentioned, each T cell is going to have what we call a T cell receptor, and this picture here is a model of the T cell receptor. This is a membrane-bound protein that contains a constant region. This constant region is going to be the same in all T cell receptors that are produced within the body. They all contain a variable region, and each of these is going to be unique to a T cell that is produced within the bone marrow. Now, how do we get this unique characteristic when we only have 25,000 genes in the human genome, and actually only one gene that encodes for the T cell receptor? Well, the uniqueness, which can produce millions of different types of T cell receptors, comes from really two different events here. First, it comes from genetic rearrangements that actually occur within the T cells as they are being produced. Secondly, it occurs through hypermutation. Together, these two events are known as VDJ recombination. We are not going to spend time talking about this as it is a very complex mechanism reserved for upper-level college courses. But I do want you to know that these variable regions do exist and that cells do have a way to generate genetic variability here within the T cell receptor. Now, this variable region also contains a region that allows for antigen binding, and so we call this the antigen binding site. And this is specifically what is going to bind to the epitope of an antigen. Now, in the next few slides, we are going to talk about how T helper and T cytotoxic cells are activated. But there is a common theme that I want you to pay attention to here, and that is that T cells really require 3 signals in order for their full activation. The first is that the T cell receptor must bind to an epitope. This is kind of like the binding between a PRR and a PAMP. Secondly, the co-receptor, CD8 or CD4, must bind to one of the MHCs. And then finally, cytokines from the cell that the T cell is binding to will help to activate that T cell. Now, any of these individually might have a very small stimulation effect, but they're not going to lead to an effect that's really going to activate those T cells. I kind of liken this system a bit to a fail-safe system. Think of the movies where you have 3 or 4 people and every single one of them has to at the same time insert a key and turn it for some sort of response to take place. Well, that's really what's happening here. Another way to think of it is like a 3-code authentication. These days, authentication using our phones as a way to confirm who we are is very common. And so you could kind of think of this as a 3-code or a 3-step authentication in order to activate those T cells. And you may be thinking, why is it so hard? Why do we need all of these signals to activate T cells at one time? Well, really, when these 3 signals are present, this is surely a sign of an infection. Any of them individually may not really signify that an infection is occurring inside the human body. Let's now dive into how T helper cells are activated, and we're also going to talk about their response or their function after their activation. In green is a professional antigen presenting cell, and in red is our T helper cell, and purple here is our T cell receptor. Now, we've already defined that T helper cells will respond or interact with epitopes that are presented on the surface of MHC2. And we've already described how MHC2 is only found on professional antigen presenting cells. Let's look at the signals that have to occur for these T helper cells to become activated. First, that T cell receptor has to recognize an epitope shown here as this orange triangle. Secondly, a CD4 co-receptor will interact directly with MHC2. So here then are 2 of the 3 signals that we need to see. CD4 interacting with MHC2 and the T cell receptor bound to an epitope. The third signal is the secretion of cytokines from the professional antigen presenting cell, which would help to activate the T helper cell. Now, it's important to note here that recall, epitopes are only part of an antigen, and when professional antigen presenting cells engulf pathogens, they engulf multiple antigens. And so this professional antigen presenting cell here may be presenting multiple different epitopes on its surface, which means it could activate a variety of different T helper cells, each that recognize a different epitope. The T helper cell then can respond in a couple of different ways. First, we have what's known as T helper 1 cells, and these are going to be involved in activating both the innate and the cellular immune response. In doing so, or how they do so, is to increase or enhance the killing of TC and NK cells. T helper 1 cells also improve phagocytosis by macrophages and neutrophils. T helper 2 cells stimulate B cells to produce secreted antibodies, which we'll talk about in a future lecture. And all of this is really a result of the cytokines that are secreted by these T helper cells. So T helper cells, regardless of whether it's Th1 or Th2, are really orchestrating the innate and the adaptive immune response by regulating the cytokines that are being secreted from it. Finally, there is going to be a subset of these T helper cells that will become part of the memory. All adaptive immune cells will produce memory cells upon their activation, and the role of these memory cells is really just to remember the pathogen or remember the epitopes that they've seen. And the benefit of this then is that if this epitope is seen again by this T helper cell, well, the response would occur more strongly and it would occur more quickly than the initial response when it sees the pathogen for the first time. All right, so T helper cells are master regulators of the immune response. Cytotoxic T cells, on the other hand, have a slightly different response. Actually, it's quite different. I shouldn't say slightly here. And the way that these cells are going to work, again, our cytotoxic T cell is going to be shown in red. The green in this case is going to represent any cell that is presenting an antigen. And again, these cells are going to present antigens on MHC1. So in this case here, we're going to have our antigen again or our epitope in orange. In purple is our T cell receptor. And again, we require 3 signals for these cytotoxic T cells to be activated. First signal is the binding of the TCR to the epitope. The second is an interaction between CD8 and the co-receptor, the CD8 co-receptor and MHC1. And then the third particular signal here, again, would be secretion of cytokines. And these cytokines may be coming from T helper cells that have actually already been activated. And so those 3 things there will lead to the activation of cytotoxic T cells. Now, upon this activation, these cytotoxic T cells are going to begin to proliferate or divide rapidly, just like we saw with those T helper cells. Some are going to become memory TC cells, and some are going to become what we call cytotoxic T lymphocytes, or CTLs. And these CTLs are killers. What they end up doing is they end up secreting perforin, which is going to create pores in the membrane of the cell, and this is going to disrupt homeostasis by allowing ions in and out across that cell membrane, by allowing for water to flow freely. These pores that get formed are nonspecific, and so this is going to start to disrupt the cell. But the other thing that gets secreted from CTLs is granzymes, and granzymes are enzymes. Specifically, they are proteases. So they are enzymes that digest protein, and these are going to trigger or activate apoptosis, which is a form of programmed cell death. And so the role of these CTLs then is to kill the infected cell, therefore preventing the spread of infection. Now, to me, pathogens are fascinating. It amazes me how they find ways to avoid detection by the immune system. And MHC1 and regulating the presentation of MHC1 on a cell surface is one of the ways that some viruses have figured out to avoid immune detection. So there are a couple of them that I want to highlight really briefly here. One is the human cytomegalovirus, which is a herpes virus. It's part of the herpes virus family. And HCMV, as it's called, encodes a protein that blocks the function of TAP. And so by blocking the function of TAP, it prevents peptides, even those from the virus, from ever being loaded onto MHC1. And what's going to happen then is that if MHC1 doesn't have peptides loaded on it, it never makes its way to the cell surface. Another virus, known as adenovirus, prevents this secretory step of MHC1 presentation. Again, this prevents MHC1 and epitopes from the virus from ever making its way to the cell surface. And so really what's happening here is that these cells that are infected with virus are being hidden from those cytotoxic T lymphocytes. Well, amazingly, our immune system has a way to detect these cells that don't have MHC1. And that mechanism is the NK cell, or the natural killer cell. And what makes the natural killer cell different from the cells that I've talked about today so far is that they are part of the innate immune system. They recognize a molecular pattern, and the pattern that they're looking for is MHC1. So let's take a look at how this would work in practice. Normally, all cells should be expressing MHC1. There are exceptions to this. For example, cancer cells may not express MHC1. And in this Now, what's amazing is that even in the presence of these viruses and other pathogens that prevent the expression of MHC1, we have a way to kill them, and that is with the NK cell, or the natural killer cell. The natural killer cell, unlike TCs and T helper cells, is part of the innate immune response. It recognizes molecular patterns, and in particular, the molecular pattern that it recognizes is MHC1. So let's take a look at how this works. In this case here, NK cells have inhibitory receptors that bind to MHC1. If the MHC1 is present, this leads to a signal inside of the NK cell to remain silent, to remain quiet. I kind of think of this like a pacifier for a baby, where if you stick the pacifier in the baby's mouth, it's going to usually put that baby to sleep or keep them quiet for a little bit. There's also a secondary interaction here, and that is between a cell marker that we're just going to leave nameless for right now and an activating receptor on the NK cell. This is providing or stimulating a positive response. Now, in this case here, as we're seeing this, MHC1's interaction with the inhibitory receptor is much greater, is signaling a much greater inhibitory response than is the activating response between the cell marker and the activating receptor. And so the NK cell in this case then would remain quiet and inactive. But let's now imagine that a virus infects this cell. It blocks the presentation of MHC1, which removes that inhibitory signal. Well, now we have a baby without a pacifier, or basically Hulk. We have somebody who's transforming from a normal inactivated cell to something that is active and ready to explode. And that explosion is the secretion of granzymes and perforin. The NK cell functions a lot like the cytotoxic T lymphocytes in that it secretes granzymes and perforin to kill this infected cell. So let's summarize these responses here to MHC1. If MHC1 is present, or if it's present and presenting self-epitopes, there's going to be no response from the cell. If MHC1 is present and presenting a foreign epitope, for example, one from a virus, then the TC cells will become activated and kill the cell that is presenting the epitope. Finally, if MHC1 is absent from the cell surface, NK cells will become activated. I want to include a really quick side note here, and that is that this particular interaction is the source of intense research because, in particular, the research focuses on cancer biology. Some cancers will prevent the expression of MHC1 on the surface of cancer cells. And so researchers are looking for ways to potentially use NK cells to identify these cancerous cells. All right, so let's just summarize some of the major points that we talked about. We talked primarily about three types of cells: the T helpers, the TC or the T cytotoxic, and the NK or natural killer cells. First, we talked about how these cells are stimulated. In this little table here, I'm summarizing for you the major histocompatibility complex that each of these cells interact with. We said that for T cells, they require three signals for activation. One, the T cell receptor must interact with a particular epitope. Two, the co-receptor must interact with MHC. And then third, these cells must respond to cytokines. T helper cells interact with MHC2, and that's an interaction that's occurring between the CD4 co-receptor on the T helper cell and MHC2 on the professional antigen presenting cell. TCs interact with MHC1 on any nucleated cell, and the TC uses its CD8 co-receptor to identify MHC1. NK cells, on the other hand, don't require the identification of a specific epitope. Instead, they have an inhibitory receptor that will bind to MHC1 on the cell surface. And we talked about the response of these cells. T helper cells will secrete cytokines to orchestrate the immune response. Cytotoxic T cells will kill infected cells with the production or release of granzymes and perforin. And NK cells will also kill cells, but specifically it's going to kill ones with no MHC1 on their surface. We hope that this summary has been helpful, and I look forward to talking to you about B cells and antibodies in the near future. Take care.